Genome sequencing is a process that decodes the genetic material of an organism or a virus. By comparing the sequences of different samples, scientists can track the spread and evolution of viruses, such as SARS-CoV-2, the virus that causes COVID-19. Genome sequencing can help identify new variants of SARS-CoV-2 that may have different characteristics, such as increased transmissibility, severity, or resistance to vaccines and treatments. These variants are classified by public health organizations as variants of interest or variants of concern, depending on their impact on public health. Genome sequencing can also help develop vaccines and tests based on the genetic code of the virus. Vaccines work by stimulating the immune system to produce antibodies that can recognize and neutralize the virus. By sequencing the virus, scientists can identify the most important parts of its genome that encode for proteins that are exposed on the surface of the virus, such as the spike protein. These proteins are then used as targets for vaccine development, either by using a weakened or inactivated form of the virus, or by using a synthetic version of the protein or its genetic code. Tests for COVID-19 also rely on genome sequencing to detect the presence of the virus in a sample. Most tests use a technique called RT-PCR, which amplifies a specific region of the viral genome that is unique to SARS-CoV-2. By sequencing the virus, scientists can design probes and primers that can bind to this region and produce a signal that indicates a positive result. However, if the virus mutates in this region, the test may fail to detect it. Therefore, genome sequencing can help monitor the changes in the viral genome and update the tests accordingly. Genome sequencing is a powerful tool for tracking and fighting COVID-19 and other infectious diseases. By using genomic surveillance, scientists and public health officials can monitor the emergence and spread of new variants, evaluate their potential impact on disease severity and vaccine effectiveness, and develop new strategies for prevention and treatment based on the genetic code of the virus. The human genome is not static, but constantly evolving in response to various environmental factors, such as infectious diseases. One of the most devastating pandemics in human history was the Black Death, caused by the bacterium Yersinia pestis, which killed up to 50% of the population in Europe, Asia, and Africa in the 14th century. How did this massive mortality event shape the evolution of our immune genes and influence our ability to fight disease today? Recent studies have revealed that some immune genes were under strong positive selection during and after the Black Death, meaning that they conferred a survival advantage to those who carried certain variants of these genes. These variants affected the production and function of proteins that are involved in recognizing and eliminating pathogens, such as Y. pestis. For example, one variant of a gene called IRAP2 was associated with a higher production of a full-length protein that helps to activate immune cells and control the infection. Individuals who had 2 copies of this variant were more likely to survive the plague than those who had different versions of the gene. However, this evolutionary trade-off also had a downside. The same variants that protected against the plague are now linked to an increased risk of autoimmune diseases, such as Crohn's disease and rheumatoid arthritis. These are conditions where the immune system mistakenly attacks the body's own tissues, causing inflammation and damage. It is possible that these variants were beneficial in the context of a severe infection like the plague, but became detrimental in modern times when we face less exposure to pathogens and more environmental triggers for autoimmunity. Therefore, the Black Death left a lasting mark on some of our immune genes, which may have helped us survive one of the worst pandemics in history, but also made us more susceptible to certain chronic diseases in the present. This shows how our genome reflects our past interactions with pathogens and how they have shaped our immune system over time. Did you know that about 8% of your DNA comes from viruses that infected your ancestors millions of years ago? These ancient viruses are called human endogenous retroviruses, HERVs, and they are mostly harmless. But some of them may actually help us fight infections by enhancing our immune system or protecting us from other viruses. One way that HERVs can help us is by activating when we are exposed to interferons, which are signaling proteins that our cells release when they detect a viral infection. Interferons trigger the production of antiviral proteins and alert other cells to defend themselves. Some herbs can bind to these interferons and turn on genes that are involved in immunity, such as genes that regulate inflammation or genes that encode antibodies. Another way that herbs can help us is by interfering with the replication of other viruses. Some herbs can produce viral proteins that block the entry or exit of other viruses from our cells, or that degrade their genetic material. For example, one HRV protein called syncytin-1 can prevent HIV from infecting cells by competing with its receptor. Another HRV protein called REC can inhibit the replication of hepatitis B virus by binding to its RNA. Herbs may also have other beneficial effects on our health and development. For instance, some herbs are involved in the formation of the placenta, which is essential for fetal growth and maternal health. Some herbs may also influence gene expression in the brain and affect neural development and function. So, while some viruses may cause disease and harm, others may have become our allies over the course of evolution. By co-opting their genetic material, we have gained new tools to fight infections and adapt to our environment. Herbs are a fascinating example of how our genome is shaped by ancient viral pandemics and how we can turn a threat into an opportunity.